Bismillahirrahmanirrahim. Assalamu alaikum. My name is Madiha Kiran, Department of Biology, AFSAC Fort Road, Rawalpindi. Students, today we will discuss the polymerization of nucleotides from chapter number 2, Biological Molecules. Students, you will be able to describe how individual nucleotide bond to form polynucleotide chain in nucleic acid. Before starting the topic, let us discuss what is nucleic acid. Nucleic acid is a biological molecule. We know it as DNA and RNA. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. DNA and RNA both are polymers. What is a polymer? Polymer are molecules which are made up of many smaller units known as monomers. Different monomers together, they combine through condensation reaction to form a polymer. So DNA and RNA both are polymer, and the monomer used in making of these DNA and RNA are nucleotides. What is the function of nucleic acid? Nucleic acid holds all the genetic information of an organism, and it also plays a very important role in many functions, just like protein synthesis. DNA has double-stranded structure while RNA has single-stranded structure. So DNA contains adenine, guanine, cytosine and thymine as nitrogenous bases, while RNA contains adenine, guanine, cytosine and uracil as nitrogenous bases. What are nitrogenous bases? Nitrogenous bases are actually organic compounds. They are heterocyclic organic compounds with nitrogen in them. Why they are called bases? Because they can donate electron to other molecules. That's why they are known as bases. There are two types of nitrogenous bases, purines and pyrimidines. Purines are actually double ring molecules and pyrimidines are actually single ring molecules. Composition of nucleic acid. What is nucleic acid composed of? Nucleic acid is composed of many monomers called nucleotides. What are nucleotides? Nucleotides in case of DNA are termed as deoxyribonucleotides. In case of RNA, they are termed as ribonucleotides. Nucleotides are composed of three units: phosphate group attached to the sugars and a nitrogenous base which is also attached to the sugar. Now the sugar in case of DNA and RNA is different. The sugar in case of RNA is ribose sugar which has hydroxyl group at 2'. The sugar in case of the DNA is deoxyribose sugar. You can see it has hydrogen ion at the 2' position. The absence of hydroxyl ion at the 2' position in the DNA makes the DNA more stable structure. While in RNA, hydroxyl group is present, which is an unstable structure. RNA usually degrades within 30 minutes, but DNA has to live a long life. It has to be a stable structure. That's why hydrogen group is present at 2', not the hydroxyl group. Now these nucleotides, they are attached to one another through a special kind of bond. This kind of bond is known as phosphodiester bond. Now look at this structure. This is sugar, this is nitrogenous base at the 1' position, and this is phosphate group at 5' position. Now this one is one nucleotide. This one nucleotide is attached to another nucleotide through a special bond known as phosphodiester bond. Today we are discussing how this phosphodiester bond is formed. Now during the polymerization reaction, formation of the phosphodiester bond we just discussed, now the incoming nucleotide should bring 3 phosphate group with it. This is the incoming nucleotide. Now look at the incoming nucleotide. It has 3 phosphate group at the 5'. This is a DNA template and this is the growing chain. Now look at the growing chain. It starts at the 5'. The sugar here is 5' at which the phosphate group is attached. And the chain is growing at the 3'. Now at the 3' of the sugar, hydroxyl group is present. Now this end of the sugar will form a bond with this phosphate group, and this kind of bond is known as phosphodiester bond. When this bond is formed, these two phosphates are released as pyrophosphate. This is a condensation reaction. During the condensation reaction, the incoming nucleotide is forming a bond with already existing 3' end of the growing chain. A water molecule is released. This water molecule released helps in the removal of pyrophosphate. This phosphate now form a ester bond with this carbon. An ester bond is formed between this 3' carbon and phosphate group. Both are linked through oxygen. Note that this phosphate is already bonded to this 5-carbon sugar here through an ester bond. That's why these two bonds are known as together phosphodiester bonds. This water molecule released in this condensation reaction takes part in the removal of pyrophosphate. Now you can see that the water molecule is released as this, and the phosphorus is now attached to 3' carbon through a special kind of bond. This bond is known as ester bond. This is another ester bond, and these two are known as phosphodiester bond. Now let's discuss the role of the water. This water helps in the removal of these pyrophosphates. In the condensation reaction, usually dehydration reaction takes place when monomers join together to form a polymer. Water molecule is removed. But in this polymerization reaction, the water molecule released is not released as a byproduct, but it is used to release the pyrophosphate. This is how the chain grows. This is the growing chain. This is incoming nucleotide. The incoming nucleotides attach its phosphate through phosphodiester bond, and two phosphates are released. Now this image shows the phosphodiester bond. You can see the phosphate is linked to 3' carbon and 5' carbon, and it is combining and uniting the two nucleotides. This kind of bond is known as phosphodiester bond. It is also termed as 3' to 5' phosphodiester bond. So this is how polymerization reaction takes place. Now let us revise. This is the DNA strand, this is the template strand, and this is the growing strand. You can see the growing strand has two terminals, 5' end and the 3' end. This is the incoming nucleotide. The incoming nucleotide has 3 phosphate groups. When the condensation reaction takes place, these 2 phosphate groups are removed with the addition of water molecule. While this phosphate group forms an ester bond with the 3' end of this sugar. You can see over here, this is an ester bond and this is already existing ester bond. That's why these two bonds are called phosphodiester bonds. So the incoming nucleotide forms a phosphodiester bond with the already growing new strand. Now notice that the growing strand has two terminals. Number one is the 5' end. This is the 5' sugar. That's why this end is known as 5' end, and it always has a phosphate group. The other end is known as the 3' end. This is the 3' end. You can see over here. It always has hydroxyl group. Students, after listening to today's lecture, you will be able to answer the following question. What is phosphodiester bond? How is it formed? How one nucleotide polymerizes with other? I hope you understood today's lecture. Thank you.